I'm very pleased to introduce speakers for Session 1. Session 1 for Challenges in Life Cycle Management of Long-Acting Injectable and Implantable Drug Products has 4 speakers. In order of presentation, Miyoung Yoon, PhD, is the acting team lead of
Hang on one moment, please.
of Research and Standard in Office of Generic Drugs at FDA. Dr. Yoon's work focuses on conducting regulatory and research activities, leveraging clinical pharmacology tools and expertise, and actively developing collaborations with multiple stakeholders. Hao Zhu, PhD, is the Deputy Director at Division of Pharmacometrics, Office of Clinical Pharmacology at CDER at FDA. Dr. Zhu's division reviews the pharmacometrics-related submissions and supports pharmacometrics-related policy development across CDER at FDA. Ameya Kohojkar, MS, RAC, is the Associate Director in Regulatory Affairs within the Sterile and Biosimilars Organization at Teva Pharmaceuticals. Dr. Kohojkar is responsible for all core development sterile portfolio comprising several complex products. Michael Fitzgerald, PhD, is the Head of Formulation, Research and Development for Injectable Products at Viatris, with over 26 years of experience in the pharmaceutical sector. Dr. Fitzgerald led CMC development progression for both ANDAs and NDAs, with focus on development of approaches supporting clinical equivalence as a drug product progresses from early to late stage pivotal clinical studies. Good morning, everyone. In this presentation, we will briefly talk about the model-integrated evidence approach from a generic drug perspective. We will talk about current challenges in the development and assessment of generic long-acting injectable and implantable drug products, and opportunities with model-integrated bioequivalence to help overcome such challenges. This is a disclaimer that this presentation reflects my view and does not represent FDA's views or policies. Today, first, we will briefly go over the challenges in bioequivalence studies for generic LAI products. And we will go over what are the opportunities with model-integrated approaches. And then we will share the regulatory experience, mainly from pre-ANDA meetings. And we will wrap up the presentation with the current gaps and open questions, which motivated us for this workshop. Long-acting injectable products are formulated to achieve extended release of drugs from days to years. This long duration of action leads to unique challenges in BE studies for generic long-acting injectables. Typical single-dose crossover study design may not be practical or cannot be recommended. This slide illustrates the challenges associated with different types of long-acting injectable BE studies. In general, if there's a safety concern, multiple dose crossover steady-state studies are recommended. With those, multiple challenges are there, most notably at the extremely long study duration. If safety is not a concern, studies can be done with healthy volunteers, but then with the parallel design, high PK variability and large sample size are also the problems. These challenges are well reflected in the currently approved ANDAS status. The majority of the long-acting injectable products don't have generics yet. Modeling and simulation approaches can help overcome these challenges regarding long-acting injectable products. Model-integrated approach, also referred to as MIE throughout this presentation, means using modeling and simulation to generate pivotal BE evidence for decision-making. Specifically, we will focus on MIE to support product approval in this workshop. Through a pre-specified model-based analysis of an in vivo BE study and simulated or virtual BE studies, MIE can support long-acting injectable product approval. For definition and further details of MIE approaches, please refer to Dr. Zhao's 2019 publication in CPT, the citation provided at the bottom of the slide. MIE can also support alternative BE to otherwise recommended in vivo BE studies in combination with in vitro BE testings. Although not the focus of this workshop today, but MIE can use mechanistic models such as physiologically based pharmacokinetic modeling. So mechanistic PBPK modeling and simulation can support alternative BE in combination with relevant in vitro studies as just mentioned. Mechanistic models such as PBPK models can integrate key formulation attributes and physiology. In this case, MIE with PBPK modeling and relevant in vitro studies are used to support product approval. FDA has been supporting model-based bioequivalence approaches for long-acting injectable products. Our GDUPA research programs have been supporting multiple external and internal research activities on this end. So external research has been supporting development of innovative MIE approaches for long-acting injectables, as well as internal research programs have been supporting the assessment of applicant proposals with MIE for LAI BE assessment. So the progresses have also been reflected in product-specific guidances. For example, paliperidone PSG knows that quantitative method and modeling, for example, such as model-integrated approach, can be used to support demonstration of BE for this product. And also another example, levonorgestrel intrauterine device. Alternative BE criteria was set up with support of the modeling. This slide summarizes the current and past FDA-funded grants and contracts involving the modeling and simulation in general for long-acting injectable products. So here are the examples of pre-end meeting experience. We have seen several innovative MIE approaches submitted by generic drug industry for long-acting injectable products in pre-end meetings. Some examples of MIE purposes include reducing the study duration and the sample size, supporting alternative BE metrics with alternative BE limits, supporting the use of less challenging sampling schemes, etc. The strategies used for those model-integrated approaches, for example, include leveraging the published population PK models for their proposed MIEs, conducting in vivo clinical PK studies that are different from guidance recommended designs to use model development and assessment, conducting in silico bioequivalence studies for bioequivalence analysis, using virtual or simulated BE studies to justify the proposed study designs or other aspects of MIEs. These reflect opportunities using model-integrated evidence approach for making bioequivalence studies more efficient and feasible, and for long-acting injectable products, and also shows that what kind of processes are in MIEs. Model-integrated evidence approach for regulatory applications. What are the considerations? This slide definitely not the full list of regulatory considerations, but what we have frequently emphasized with these examples. MIE should be demonstrated that it is sensitive to detect formulation differences with confidence. And for the intended regulatory purposes, sufficient validation and verifications are needed. Again, MIE needs to be capable to discern formulation differences with appropriate type 1 error control. What is the most important from regulatory perspectives? Modeling analysis plan is needed and should be pre-specified. Given the complexity of the model-based analysis, this may be challenging. This slide shows some thought on current advantages and challenges in MIE based on our experience, both regulatory and research. Clearly, model-integrated evidence approaches have promises to help address current challenges in LAI-BE areas. It can allow us to make LAI-BE studies more feasible while ensuring confidence in the decision. For example, it can make study duration much shorter. We note that clarity in validation and verification of models and also the approaches for the purpose of MIE is needed. MIE has been discussed multiple times, including this year's GDUPA Science and Research Initiatives public workshop. These are some of the topics discussed regarding MIE for long-acting injectables. These questions reflect the high interest from multiple stakeholders about the possibilities with MIE to reduce regulatory barriers for generic long-acting injectables product. Again, these raised questions reflect opportunities how MIE can help address the current challenges. I would like to wrap up this presentation by noting that we acknowledge the key challenges in applying MIE for long-acting injectable bioequivalence include clear consensus on the acceptable model validation and verification for MIE applications, and also acknowledging MIE as BE approaches and methodologies and validations on that end. We believe it is time, and FDA and CRCC have taken the initiatives to develop and establish best practice in MIE for bioequivalence with you all through this public workshop. Thank you for your attention again. With that, I would like to acknowledge my colleagues in the QMM and ORS, especially the quantitative clinical pharmacology team. And also, thank you, our external collaborators.
I would like to thank the organizer for providing me the opportunity. So I'll be happy to share some of our experiences by using modeling simulation to support the use of non-acting antipsychotics. I have a disclaimer. The views presented here are my personal views and may not reflect the position of US FDA. I'm going to open up with a background and introduction. Then I'm going to share some case examples on how modeling simulation is used to support the development of non-acting injections. Then I'm going to conclude with some take-home messages. Psychiatric diseases such as schizophrenia and bipolar I disorder are severe, debilitating mental disorders. Chronic treatment is essential to prevent relapse and to control symptoms. However, compliance is a common problem in this patient population. Therefore, long-acting injections have been developed in order to improve compliance and patient care in recent years. And this table summarizes some of the examples of the marketed non-acting injectable antipsychotics. For example, risperidone non-acting injection is given once every 2 weeks under the trade name of Risperidone Consta in the United States. Paliperidone can be given either once monthly under the trade name of Invega Sustenna or once every 3 months under the trade name of Invega Trenza. Likewise, aripiprazole can be given once monthly under the trade name of Abilify Maintena, or given once monthly, once every 6 weeks, or even once every 2 months under the trade name of Aristata. Now I'm going to focus on how modeling simulation can be applied to support non-acting injection development. It is an essential tool to optimize dosing regimens, to define dosing windows, to select the reinitiation plans, and to adjust the dosing regimens in patient subgroups. Well, let's focus on the Invega Sustenna, which is a paliperidone non-acting injection. The product is approved for the treatment of schizophrenia and schizoaffective disorder in adults. Due to the particle size and low solubility, the product demonstrates slow dissolution. As shown in the PK profile, the median maximum concentration is achieved on day 13 after dosing. The apparent half-life varies from 25 to 49 days. As demonstrated in section 14 of the product label, the sponsor conducted 4 clinical trials with the tested dosing regimens listed in the table. Study 2 to 4 are conducted with only maintenance dosing regimens given once monthly. Study 1 has a loading dose of 234 mg given on day 1 and followed by monthly maintenance dosing. This table summarizes the treatment effect following the 4 clinical trials. Almost all the dose arms demonstrated superiority over placebo by the end of the treatment duration. However, the approved dosing regimen in general patients is different from any of the dose tested in the trials. In fact, the approved dosing regimen contains 2 loading doses, a dose of 234 mg on day 1 and followed by 156 mg on day 8. Then the dosing is switched to maintenance dosing given once monthly. The simulated PK profile for patients without prior treatment is shown in the figure. The blue band represents the target exposure range. The red solid line shows the PK profile following the approved dosing regimen. The exposure will achieve the target exposure range within a week, yet the highest concentration achieved by the end of the 2 weeks are still below the concentration considered tolerable as shown by the black dashed line. The figure also illustrated the PK profiles following 2 alternative dosing regimens without additional dose on day 8. The concentration may reach desirable range much slower. And this figure summarizes one additional scenario when patients are stabilized with prior treatment on paliperidone. All simulated PK profiles start from the lower bound of the target exposure range. Still, as expected, the approved dosing regimen allows a quick concentration rise to the target exposure range, yet still below the tolerable concentration range. The additional dose on day 8 is beneficial compared to other alternative dosing regimens. Now we are moving on to a second scenario. We understand that sometimes patients are unable for injection exactly on time, for instance, due to holidays or weekends. It is useful to establish flexibility on the dosing window that makes the injection more practical. The product label shows a dosing window of 4 days for the second injection and plus or minus of 1 week for maintenance dosing. Again, this dosing window effect was assessed by using Miyoung simulation. The red line is the PK profile following the scheduled dosing. The 2 dashed lines show the PK profiles following the dosing 1 week earlier or 1 week later. We can see the concentrations still fell into the target exposure range to a large extent within the dosing interval. We understand that compliance is the biggest issue in this patient population. Using non-active injection can hopefully improve patient compliance, but there is still a chance that patients may miss dosing. The label provides a detailed strategy to handle patients with different durations of missing doses. So I'm going to share one example that a patient discontinues for less than 4 weeks since the last injection. The figure to the left shows the concentration decline over time if the patient misses the dose. However, the figure to the right shows the concentration change following the recommended dosing table, and you may see the concentration climbs back rapidly. Using the drug in specific patient population with compromised renal function is always an important issue. However, the study in patients with compromised renal function might not be necessary using the non-acting injections. Instead, we can use the clearance change in patients with compromised renal function using oral formulation to derive dosing adjustment strategy for the non-acting injection. By factoring in appropriate clearance, we can come up with dosage adjustment in patients with compromised renal function for both loading dose and maintenance dosing with no need for unnecessary clinical trials. With that, I'm going to conclude today. I have the following take-home messages. Modeling simulation are essential tools to facilitate development of non-acting injectable products. Essentially, it can be utilized to support new dosing regimens, to define dosing windows, to select reinitiation plans, to adjust dosing regimens in subgroups. So I want to really thank Dr. Yanni Wang and other OCP and medical colleagues. I also wanted to thank you, our audience, for your attention. Thank you very much.
Hello, my name is Ameya Kohojkar and I work as an Associate Director at Teva Pharmaceuticals in their Regulatory Affairs Department. Today, I'll be presenting on industry perspective, regulatory challenges in development of generic long-acting injectables. I'd like to start by thanking the CRGC group for this initiative and giving me an opportunity to present. Before I present, I would like to read out a disclaimer. The opinions expressed in this presentation are mine and not necessarily those of Teva Pharmaceutical Industries or its affiliates, collectively known as Teva. This presentation has been prepared for discussion purposes only. Neither Teva nor any of its employees or representatives make any representation or warranty, express or implied, as to the accuracy or completeness of any information contained herein. The information and examples presented originate from individual experience and may not represent the full scope and/or examples of Teva. Nothing contained within the presentation is or should be relied upon as a promise or representation as to the future, and Teva expressly disclaims any obligation to update the information if it should change. First, I'd like to give some background on long-acting injectable LAI formulations and the topics that I will be discussing during the presentation. LAIs are formulated to achieve an extended drug release action from days to months. LAIs have many advantages for patients. It provides relief from needing to remember to take medication daily. It typically maintains therapeutic levels of the drug for extended periods and may reduce the risk of non-adherence to daily dosing routines. FY 2016 Regulatory Science Report noted LAI formulations include biodegradable injectable microspheres and in situ gelling implants. Compendial in vitro release methods for these complex formulations are not well developed, and demonstration of BE for these products can be challenging. Since then, FDA has published product-specific guidances (PSGs) for several LAI formulations, all requiring an in vitro and an in vivo study. There are numerous challenges in both in vivo bioequivalence studies as well as in vitro studies. I will elaborate more about these challenges in the upcoming slides. Speaking about challenges in vivo BE studies, some products need studies in patients, as an example, antipsychotic medications. This makes recruiting difficult. To ensure there is an appropriate sample size, enrollment happens at multiple clinical centers. These centers are often located in multiple countries, resulting in an increased complexity of clinical trial applications to local authorities if needed. Data management, safety management, reporting for such studies is also difficult. Rare or orphan drug indications make recruitment much more challenging or not feasible at all. Another challenge in the longer duration of studies with potentially large sample sizes leads to significant impact or may lead to a significant impact on submission timelines. There could be occasions when all required CMC data is available and the submission is only waiting on the BE study data or results. This may result in lost opportunities like missing NCE-1 date. Most LAI formulations have complex dosing procedures. Most of the RLD products are commercialized as kits, which makes dosing complex and may require additional training to the clinical site personnel. Instruction for use or IFUs need to be followed. Reconstitution needs to be done appropriately and accurately. Any deviation may lead to an increased risk to the study or may lead to protocol violations. There is also a cost implication. Since there are multiple sites for patient trials, the cost of the RLD required for dosing and retention of the samples may increase. Different countries may have different import requirements for samples. Overall, significant resources and time is required to manage these complex BE programs. Moving on to challenges in development and implementation of in vitro methods. Developing and the need of real-time dissolution methods. The sampling time point for the dissolution method run is based on the dosage, which means the product cannot be released immediately after manufacture. The last sampling time point may very well be after a couple of days, maybe 28 days, 42 days, etc. Depending on the nature of the API, there could be extensive degradation. Based on summary basis of approvals, SBOAs, some products need to be subjected to two dissolution methods, real-time and accelerated, thus increasing the time for development and validation of the methods. Then, there is use of non-compendial apparatus which adds to the complexity. Qualification of the equipment, maintaining temperature control, and validating such systems is time-consuming and significantly increases the time and resources required in the development and validation. The dissolution method also needs to show discrimination to differentiate drug products manufactured under target conditions versus formulations with meaningful variations, and this has been one of the most critical challenges. Multiple critical process parameters (CPPs) and/or critical material attributes (CMAs) may need to be changed simultaneously to see a different release profile. The change in the parameters may sometimes need to be more than the FDA recommended plus or minus 20% of the target. Establishing an in vivo in vitro correlation (IVIVC). Development of 1:1 correlation may be difficult to establish. There is a lack of compendial in vitro release testing methods, and LAI formulations typically have an initial burst and multi-phase release mechanism, making demonstration of IVIVC even more difficult. Well, these were the challenges. Moving on to potential next steps. From an applicant or a generic manufacturer perspective, understanding FDA expectations for model integrated evidence (MIE) in an ANDA is extremely important. It would be beneficial to both industry and the FDA if there was a mutual understanding of the information to be submitted in pre-ANDA meetings to make the most of the meeting. The meeting is generally only an hour long and typically there are multiple other questions to discuss. Thus, having a clear understanding of requirements would help the sponsors prepare better questions for such meetings, making the most efficient use of time. it is important that there is also an understanding of the validation requirements and questions be asked during the pre-ANDA meeting. And most importantly, generic manufacturers need a clear roadmap to approval following the MIE approach. Model integrated evidence approach is currently in its nascent phases, and its development, like some other in vitro BE techniques, will only help the generic industry to bring LAI products to the market sooner. And there has been progress in this direction. As an example, the PSG for paliperidone palmitate, published in August 2021, encourages the applicants to consider quantitative methods and modeling, for example, model-integrated approach, to support demonstration of bioequivalence. This is very promising for prospective applicants. In summary, there are several challenges when developing LAIs, right from developing a Q1 Q2 formulation to completing a successful BE study. BE studies and dissolution method development are complex, time-consuming, and expensive. MIs may help accelerate availability of generic LAIs. Currently, there are no standard expectations on the data needed to be included in the pre-ANDA meetings. It would be very beneficial if available recommendations for specific products are included in the PSGs. Lastly, it is very important to establish a working partnership between academia, FDA, and the industry to advance the development of innovative techniques to help make generic alternatives available to public. I would like to thank all my colleagues who helped and supported me to put this presentation together. Thank you all for your time and attention.
Hello everybody, my name is Michael Fitzgerald and I'll be presenting an industry perspective on incorporation of BE modelling and long-acting injectable development challenges. The views expressed in this discussion are based on broad industry commentary and should not be interpreted as unique or specific to BIOEQUIVALENCE Incorporated or its subsidiaries unless specified. So covering the topics on the agenda, first item is introduction. I'm very pleased to join today's discussion on model-integrated approaches for complex generic products, including long-acting injectables. Viatris supports CRCG's mission to enhance research collaborations with the generics industry to ultimately increase access to safe and effective generic drugs. We recognize that complex generics pose unique challenges and are amongst the drugs that bring the greatest value to patients. As a little bit of background on Viatris, the company was formed in 2020 through the combination of Mylan and Upjohn. Viatris brings together scientific, manufacturing and distribution expertise with proven regulatory, medical and commercial capabilities to deliver high-quality medicines to patients in more than 165 countries and territories. We will also cover typical considerations for long-acting injectable development, how PK modeling could assist with bioequivalence clinical studies, post-approval studies, and general scenarios and practical development challenges of adopting a PK model. We will then cover a summary of the key topics at the end. So taking typical considerations for long-acting injectable development, first of all looking at the medicine, then the patient, then how these combine to impact the study design. So first of all, with the medicine, we have complex dosage forms and processing, typically looking at technologies like nanosuspensions, microspheres, polymeric gels. These dosage forms often have multi-phase drug release, where you could find there is a burst, a lag phase, followed by a main body of release. And they have many interconnecting critical quality attributes, critical material attributes, and critical processing parameters. When I then move to the patient, you can often find you're dealing with potent drugs. The disease area requires patient use in clinical studies rather than volunteers. You have to maintain therapy for the patient. You often have a long-acting multidose to achieve steady state. And this creates challenges for studying single-dose patients. And it also creates challenges if you want to look at IVIVCs, where you have dosage forms that may differ from the target profile. When I bring all these parameters together into my study design, what it will do is it means I'll have higher patient numbers and longer studies. So now looking at how PK modeling could possibly benefit long-acting injectable development. So first of all, if we look at a conventional study approach, where I assume I'm recruiting patients, which takes longer, and I also have clinical considerations. For example, is it possible to maintain a therapeutic dose with a single injection? Or do I have to have multi-dose injection to achieve steady state and maintain therapy as well? Generally, what you can find in long-acting injectables is that you're looking at patients and multiple injections. So therefore, you'll have higher costs, longer time frames, which means it's going to take longer to develop the medicine, which can mean less access to the medicine for the patient. If we then look at the scenario where we take the study approach and we combine PK modeling with the conventional thinking, that can benefit in terms of maybe smaller studies and help with patient recruitment and overcome some of the clinical consideration challenges that we highlighted. Then if you achieve that, you're going to reduce your timeframes and costs of the studies, which means potential for earlier development approval and can lead to more access of the medicine for the patient. So now looking at PK models and their areas of potential benefits. So if we look at the development of the product and you're using PK modeling, it can help you to select the lead based on simulated data. When it comes to clinical study design, it can help you potentially minimize the size of your study. If you're approaching an IVIVC, then you could have challenges with how you do that study, and there are ways that maybe can help there. When it comes to supporting specifications and when you move into life cycle management, these are all areas that you can target with PK modeling to enhance your development and overcome some of the challenges highlighted. So taking some conventional BE study approaches, and I'm highlighting 3 schematic scenarios with increasing complexity in study design and how these impact the overall development cycle. So in the first case, example A, where I'm just looking at volunteers in a single dose, if I perform an animal study, pilot study, followed by EBE study, which then leads to submission, review, and approval. Then by moving to scenario B, where I'm dealing with patients, I'm going to extend the pilot study and BE study as it will take more time for recruitment using a single dose. And then if I'm moving to scenario C, where you're dealing with both patients and multi-dose, this will become your longest and most complex development program. So now looking at the long-acting injectable PK model build and validation, where there is some precedence from existing LAIS. And I just have a representative scenario here where I've represented phase 1 and phase 2 studies followed by phase 3, which then leads to submission, review, and approval. And there's possibly some phase 4 studies or post-approval changes that could follow after that. During the phase 1 and phase 2 phases of development, the opportunity exists to develop a PK model utilizing the clinical data that's been generated. And this can be used to support decision making for development choices of formulation or clinical design. Likewise, when the phase 3 data becomes available, you have the opportunity to validate that PK model by comparing the predicted and observed data sets. And then it can be used in the submission to support label claims for different dosing regimes, or it can be used post-approval for further activities that follow there. So now looking at long-acting injectable PK model build and validation and how that can translate to an ANDA. The first consideration I have to make is that I will have a smaller clinical data set for an ANDA development compared to an NDA. And these two representative scenarios look at how that data could be utilized for PK model build. So in the first case, I'm looking at a pilot study for multi-dose, followed by a B study multi-dose. Assuming that's successful, it leads to a submission review, then a registration approval, and then post-approval changes will follow after that. Now, looking at when I'm doing the pilot study, this offers the opportunity to build a PK model with the first set of clinical data that will become available. And then you have the opportunity to validate that same PK model as you would have the B study data, allowing you to compare the predicted and observed data sets. Likewise, with a single dose, the same logic could follow, and potentially this would offer you an easier and better pathway to build your model. There is also another scenario where you might choose to validate your PK model in advance of a B study. And in that particular case, you just need to weigh up then, based on clinical data needs, clinical considerations, is that advantageous to your development or not? If we now look at potential benefits for IVIVC approaches using PK models. So in this scenario here, if I'm looking at a potent drug, it requires multi-dose injections to achieve steady state. I could have Q and Q2 that's matching. I may have some very subtle Q3 differences. I'm looking at an RLD that has high intralot variability. I have a discriminatory dissolution test. And if you decide that a biopredictive dissolution test is useful for this product, then I will require an IVIVC. And that traditionally would involve having a target release profile, generating a slower release profile product, and also a faster release profile. Once I'd have that, I would then take it into a clinical study and hopefully build an IVIVC relationship through convolution, deconvolution between those two data sets. If I have developed a PK model, the opportunity exists that maybe your PK model could also demonstrate the same relationship, and then that gives you a potential extra utilization of PK models. Now, considering what has been discussed about some of the challenges around doing single dose and multi-dose, it may not be so straightforward to run an IVIVC with formulations that are faster and slower, when you have to factor in therapeutic needs of the patient, and if you're also in a multi-dose scenario. And this brings in the interesting scenario then, where if you have developed a PK model, do you have the option to move towards simulating the faster and slower release profiles from your in vitro data and generating a virtual IVIVC? So now coming to the key summary points. We've looked at how PK modeling can offer the opportunity to overcome some of the long-acting injectable specific development challenges. We talked about some of the practical factors, setting up studies and recruiting patients as opposed to volunteers. And we looked at the clinical considerations, weighing up the need for multi-dose, single dose, and maintaining therapy, all of which contribute to extending development timelines and increasing the costs. We also seen how PK modeling can support the improved drug product lifecycle management, which can be highly advantageous. There's also going to be need to balance the clinical data requirements to support the PK model development and validation, and comparing that and weighing it up against the conventional clinical study design that you would use to demonstrate bioequivalence. We've also touched on how PK modeling capability can support your lifecycle management. For example, there's a scenario where you may be able to use for your clinical study support using a PK model simulation to develop IVIVC approaches. And if that's possible, then it gives you the ability to manage your post-approval changes to materials, processing, and specifications, which are also very important aspects of lifecycle management of any product. And finally, I would like to thank you for your time for listening to this presentation. and I hope you enjoy the rest of the workshop.
Thank you so much for the speakers from the opening and welcome remarks, as well as the first session today. So we have a short break. So thank you for your attention, and please come back to the workshop for the session 2 by 10:05. Thank you and see you soon.